We would now be starting with our next session, which is on homeopathic drug development and regulatory innovation. I kindly request the chairs of the next session, Professor C. Nayak, Chairman, Homeopathic Pharmacopoeia Committee, and Dr. Anil Khurana, Deputy Director General, Central Council for Research in Homeopathy, to please join the dais. I also request the speakers of the session, Dr. Rajesh Shah and Dr. Harald Hamre, to also join the dais, please.
Hello, good afternoon to everybody. Due to extreme shortage of time, we have to limit the speech of all the, I request the speakers to limit their speech. So first speaker is our young scientist, Dr. Rajesh Shah, who has been striving since a long time to, he's strongly, sincerely pursuing for the development of a new drug, and he has been working on these nosodes also, other drugs also, and he has been in collaboration with so many reputed institutes of Bombay. And he has also produced more than 10 papers, maybe 12 papers, research papers on this area. And he has also presented his ideas through different fora. Now I request him to make add some further inputs today.
Thank you so much, respected chair and delegates from all across the world. Thank you so much for creating this forum. I will talk about the new drug discovery, NDD, the challenges, solutions and opportunities, and inventors' perspective. Being a researcher in the area of homeopathy, as a homeopathy practitioner, I've been involved developing new homeopathic medicines for almost 18 years and have worked on developing several medicines. Just to give some examples, capsaicin and dihydrocapsaicin, found to be quite effective for day-to-day use as an anti-arthritic, anti-inflammatory medicine. A lot of studies have done. All the examples I'm giving, we have done preclinical and clinical trials and animal studies, in vitro model studies, etc. All the published papers in peer-reviewed journals. Hepatitis C Nosode. This was one of the first biological substance made in last several decades, and with a lot of efforts we could develop this nosode, which has shown efficacy against hepatitis C. Not only that, but it has also demonstrated anti-cancer efficacy in the in vitro model. HIV nosode, again a big struggle, and finally we could get through. It has demonstrated anti-HIV activity in an observational study of 30 patients, and it has been found clinically useful in a range of autoimmune diseases, various diseases in fact. The papers are available, and in one of the recent studies it has shown anti-cancer properties in the in vitro model. The research has been done by a very eminent molecular biologist. Hydroquinone, a chemical-based new homeopathic molecule, has shown anti-vitiligo activities, which is something which can be used by practitioners in this country, as 8% of the Indians suffer with vitiligo. I'll talk about the researchers' challenges, basically. So, if you look at the background, systematic and scientific drug discovery in homeopathy is not much explored. It is like a virgin island, more or less, especially when it comes to the biological substances. So as a result, the required regulations have not been well defined for new drug discovery. And as a effect of that, again, the research has not happened. So it's both are interconnected. And as a result, more research in this new NDD has not happened. The outcome of this is that I've worked over 100 new homeopathic medicines in last 18 years, and all these medicines are just left with me, and I have not been able to bring it to the profession. And that is one of the big challenges that I face personally as a homeopath and a researcher. So there is a need for a serious and urgent revamping for the policies and regulations to not only support the research what I have done, but to open up the windows for more researchers in many parts of the world. So what are the basic challenges? See, homeopathy being, as we all know, is not just science, but it is science and philosophy based on individualization. So the RCTs, the randomized controlled trials, is very difficult to do as a part of the research. That's the reason we have lack of new drug discovery. The conventional approach which is being applied is not quite valid so much for homeopathy, which is basically looking at the toxicity and efficacy, besides the safety. When it comes to homeopathy, because of the nanoparticle based or ultra dilute format, the efficacy check is the most important thing. The toxicity is less important. So we don't require possibly stringent toxicity criteria for evaluating the homeopathic medicines. Of course, the safety and the legality, they require attention. Currently, as Dr. Dinesh spoke very well, our guidelines are all based in drugs and cosmetic acts, around 2DD and 184, etc. It is all fragmented. There is no clarity. So there is no clarity about how to introduce a new homeopathic medicine. Since yesterday, I've been talking to various experts in this room from India and outside, and I found and reconfirmed, unfortunately, that there is no clarity in any part of the world about new drug discovery. What is the need? So the basic challenge is that we require an updated guidelines for new drug discovery. What is the solution? We require clear-cut guidelines. Step-by-step roadmap is required about the sources in terms of standardization and the reproducibility of homeopathic medicines. As we all know, most of our homeopathic medicines, especially in the biological source, are not reproducible at all. We have been using the back potencies, which were made over 100 years. We require effect check in terms of physiological effects, toxicological effects, microbial effects, and proving. Proving cannot be the only major criterion for examining the efficacy of homeopathic medicine. Efficacy check in terms of human trials, in vitro, animal model, etc. Of course, legal aspects, ethical aspects, and safety check, these are some of the things to be looked into, which can be very well organized. When we talk about ethical issues, there's another challenge. In India, when we conducted HIV clinical trial, for example, we had to struggle for 2 years to get ethical approval. There is no ethics committee in place which will accept a homeopathy research project. So we had to constitute institutional ethics committee based on, of course, ICMR guidelines. But the whole thing takes time. So if the government has the ethical committee which can take up research projects for homeopathy, that will become a boost to the researchers. So as already said by previous speakers, we need a parallel bodies like DCGI and state level FDA for homeopathy. All in all, in brief, we require a single window solution, especially for new drug discovery, a body with all necessary powers, so that we don't have to run from place to place for drug discovery. Last, there are ample opportunities. We have been all talking about only old homeopathic medicines for the last 2 days. We have not touched the scope and the potential and the opportunity behind new drug discovery. The similar principle is already established. The proven anti-disease activities of various diseases, various remedies have been already established and demonstrated. Having said that, there is a need for revamping 200 years old medicines that we are using. I know it might look like a big task, but I believe with the infrastructure of 200 homeopathic colleges in this country, it is not difficult to revamp homeopathic medicines. Dependency on back potencies, this requires to be overhauled. As a science, we cannot have a method or a system which is blindly based on some so-called back potencies. I'm sorry to say this. Some of the pharma people might not like this idea. But we are using homeopathic medicines of which we have no idea what is the origin. There is no tracking record of the homeopathic medicines, especially of biological sources that we are using. The tuberculidum nosode, made in 1875, we are using it even today from the same source material, which was made by Swan and somebody else, which was passed on from one country to another country. And please remember, in this time there were 2 world wars. And through these 2 world wars, these back potencies have survived. How? Why can't you question this? And against that as a solution, it is very much easy to reproduce the nosodes. We have already done it. We already produced Mycobacterium tuberculosis nosode at a very eminent institute of Hepkin Institute in Mumbai, which makes BCG vaccine for this entire country, which is standardized, characterized, and developed. So we need to look at our existing scenario in a more pragmatic manner and need to revamp a lot of things. There is a huge IPR opportunity, patents and intellectual property rights. We have not touched that. Hardly any homeopathic medicines except oxylococcinum and some more which have been awarded patents. I don't know whether I should say this or not, but I hold around 15 patents in India, USA, Germany, Australia, South Africa. And nobody knows about that. It is a credit brought to the India. This is intellectual property rights which you earned already, which are lying unutilized, unattended. We have even exhausted some patents. I have discontinued some patents because as a researcher, as a homeopathy researcher, I can't afford to keep paying for patents. You understand my agony? But this is lost to the country, I must say. There is a profound opportunity, last sentence, in introducing new substances for a range of new diseases and the current diseases that we have. Thank you so much for your attention.
Thank you, Dr. Rajesh sir, for your nice presentation, and appreciate your anxiety and apprehension also. For your kind information, I think he has been running from pillar to post for last couple of years for this particular issue. Being the chairman of the Homeopathy Pharmacopoeia Committee, let me clarify some of the communication gaps. Communication gaps means yesterday Dr. Manchanda also presented some that steps to be taken. That is that we have to go to the subcommittee of DTAB. Then from that we have to go to DTAB, then to the Ayush Ministry. If required, we have to go to DCGI. That is for the inclusion of the new drugs. Now coming to our domain, that is Homeopathy Pharmacopoeia Committee. We had both DGC, CRH and myself, we had long discussion with the chairperson of Professor Handa about this particular issue for a long time, and he has cautioned us. Yesterday also he admitted that some riders he put that we must be very cautious while preparing the medicines of microbial origins, nosodes particularly, and potentizing the allopathy drugs. And what we have done from the domain of HPC, we have formed the subcommittees consisting of zoologists, chemists, botanists, and other experts. And they have already given their SOP to determine the SOPs how to introduce the new drugs. And those recommendations will be put up before the next committee of the HPC, Homeopathy Pharmacopoeia Committee, that the DG of the CCRH has to do it, and after that is accepted, we have to go to the highest committee, that is scientific body, for acceptance. That is about the second aspect. In the meantime, the HPC and with the active interest of Dr. Manchanda, we have constituted so many subcommittees, the most important being the nosode subcommittee, having the experts from ICMR microbiology, from Center for Cellular and Microbiology, that virologist, then Punjab University, then the National Institute of Virology, Pune, the virologist, and they have recommended some of the SOPs, how to go for how to revamp that procedure already mentioned in the 4th volume of HPI about the nosodes. They have made some drastic recommendations how to change the concept of nosodes preparations in HPI. That is a very good step we have taken, particularly Dr. Manchanda, I must congratulate him. Another thing, whatever great thing Dr. Shah has done is a personal level, we must appreciate him, but simultaneously, I think we will get better recognition if we involve CCRH. And CCRH, under the dynamic leadership of Dr. Manchanda, he has already moved to the NIV, National Institute of Virology, Pune, and he has got the dengue nosode. And he has given our friend Dr. Surendra Singh is probably in the audience. He has given this nosode for preclinical trial. This is going on. Second thing, we have given to Punjab University for malaria, for leishmaniasis study, and for intramural research at our RRI Kolkata on JE virus. So we are not sitting idle for new drug delivery, for new drug development. Already Homeopathy Pharmacopoeia Committee and this CCRH HPL scientists, they are already in active consideration of this issue. But at the same time, I have some guidance to Dr. Shah, because he is very dynamic. Let us learn something from the past. When you gave funds to the IIT Delhi to take up a study on nanotechnology, then that one of the scientists, he did it, he wanted to publish it, and he requested the PI, his head of the department, to be a co-author. He refused. That was first instance. Second instance, when he went to the School of Tropical Medicine, and that to study the effect, prophylactic action of belladonna, that had beautiful action, in vitro and in vivo model, and that had got international publication in the peer-reviewed journal and all the regional papers and national newspapers, and that was also ridiculed by the director. And the last one is our Hyderabad experience on HIV/AIDS, and the institute, which is IICT, which was involved in the beginning, now they are withdrawing. But thanks to Dr. Shah, he has taken the help of so many institutes. But I will request him, at the end, he should not be frustrated that if this reputed institute, just like IICT, just like IIT Delhi, they should not withdraw. And even I am told that the CCRS proposal for HIV/AIDS nosodes, that is also in trouble with the NACO. So with all these past bad experience, I request you, you are very dynamic, I appreciate your dynamism. Please let us learn from the past things, and please appreciate the steps taken by CCRH and HPC to come to a conclusion. Thank you.
Thank you so much.
Excuse me,
Second.
can I have one question to Dr. Shah?
No, the discussion will be later. We really have to go on now, please. Thank you.
Okay.
Yes, we have the second speaker, Dr. Harold Hamre, who will be speaking on Anthroposophic Medicine. Yes, please.
Do I move them down with this one? I do, yes. Thank you to the organizers for inviting me to speak at this very exciting conference. And thank you to the government of India for expressly endorsing medical pluralism. Thank you. Whole medical systems, such as homeopathy and anthroposophic medicine, employ large numbers of medicinal products. According to the literature that I could find, please improve if you have better data, up to more than 1,000 substances are used in these systems. As we heard yesterday from the French representatives, often individualized in many different combinations. And as a result, it is not feasible to conduct clinical trials for each product and therapy option. It cannot be done. Accordingly, efficacy documentation for marketing authorization or registration of medicinal products from these systems often has to rely on bibliographic evidence only. And this is a structural disadvantage for these medical systems compared to systems with small number of therapy options, such as conventional medicine. But there are other types of evidence apart from clinical trials and bibliographic data. Some speakers have already commented on this, and I will discuss 3 types of evidence that have been worked on in anthroposophic medicine, both methodologically and practically. As someone previously said, anthroposophic medicine uses a large portion of the products are produced like homeopathic products. So there we are brothers and sisters, and the other parts are more similar to Ayurvedic or Unani or TCM medicine products. Single cases and case series can sometimes yield evidence for drug effects. an example from conventional medicine. 9 years ago, one case report of dramatic effects of a beta blocker, propranolol, for a baby with severe haemangioma blocking the right eye was published in the New England Journal of Medicine. The drug had been given because of concurrent obstructive hypertrophic cardiomyopathy, and the effect of this haemangioma was accidental. Other similar cases were published, leading to a change of treatment for this condition worldwide. So, why did people think the drug caused the remission of the haemangioma in this patient? Well, first, the dramatic effect was much stronger than expected effects from the steroids already given for the condition. And the effects were not explicable by the projected natural history, which was known. And second, there was a very close temporal relationship between onset of treatment and onset of improvement. Third, there was no other new treatment introduced. And there was, fourthly, even some pharmacological plausibility, since propranolol works on the blood vessels, which are affected by the haemangioma. Here you have another case with a marked effect and a close temporal relationship from a prospective study of a potentized anthroposophic product injected locally for acute pulpitis, where the pain, which had been ongoing for 13 days, remitted very soon after the injection. But here you are less certain than in the former case of the haemangioma, because you have alternative explanations for the improvement, including psychological effects as well as possible physiological effects of saline water injections. But that may be less relevant from the patient perspective. Another criteria when studying effects in single cases is, we call in German, Gestalt Correspondence. So here the gestalt, or the shape of sunburn protection, corresponds to the area covered by the sweatshirt used in the sun. So we can see, and I think you will agree with me, this is no placebo effect, and we don't need control subjects to confirm the effect in this gentleman. Yet another one. Here you have functional morphological correspondence. You inject a local anesthetic targeting the 4 sensory nerves of the toes or fingers, and the result is anesthesia of the corresponding area of the sensory innervation. Now here, the judgment of the drug effect draws on several types of information: the exact location of the sensory nerves, sense physiology, pharmacological effects of the drug, known in general, and the exact area of sensory innervation. And if it doesn't work fully, you can add a little more anesthetic to assure the effect. So the judgment is not just for scientists like me, but it's also for clinicians who use these principles when they actually guide their treatment. And here we have good old dose-effect correspondence. In this case, an inverse relation between the serum concentration of a specific drug and the symptoms of specific condition, in this case, catatonia. Now, assessment of effects in individual patients has always been part of medicine, but was neglected in the 20th century due to a focus on clinical trials. Now, my colleagues at the Research Institute have been active in clarifying the epistemological underpinning of the method and in classifying and grading the criteria. Some criteria are strong, some are weaker. Writing guidelines for case reports, which have also already been, I have heard, implemented for homeopathic case reporting by Robert van Hasselen, and by training physicians and therapists in case reports, just like you've been doing, Robert. And also there have been publishing case reports of anthroposophic treatment in the peer-reviewed literature reflecting these criteria. The second methodological tool is an upgrading of old-style narrative reports of experiences with medicinal products. Now, to clarify, in anthroposophic medicine, the homeopathic proofs play a very little or no role. So it is these old reports of, I have used this drug for this condition and it worked well in my patients, that sort of level, which you often find in old textbooks. So the Vademecum project, Vademecum is Latin for walk with me, and that means a handbook. And the Vademecum of anthroposophic medicines was conceived as a practical handbook for physicians starting to work with these products. So the book in its third edition is based on the documentation by more than 200 physicians from 18 countries, involving about 2,200 pre-structured questionnaires on indications, contraindications. We're lack of time, so perhaps you read faster than I speak, so I just let you read that and conclude. The third edition covers 827 products with more than 1,500 indications. Important is it is not just written reports, but these are critically assessed by an independent panel of experts. And they also expressly encourage people to report negative experiences to avoid bias. Now, these Vademecum indications, of course, have much lower validity than indications supported by clinical studies, no doubt. But their validity is clearly higher than much of what you find, unfortunately, in old bibliographic literature of these medicines. And, very important, the Vademecum project can capture more products and indications than clinical studies will ever do. And even high-quality case reports, you cannot do that for tens of thousands of therapy options which you have in a system. They have to be representative. Therefore, for many anthroposophic products and indication, this Vademecum collection is currently the best available evidence. The third tool is the assessment of drug effects on the level of the therapy system, in so-called system evaluation study, an example of which is the IIPCOS study of acute respiratory and ear infections, defined here on the code of 5 symptoms. The IIPCOS Anthroposophy study comprised 2 arms with more than 1,000 patients and choosing treatment by anthroposophic or conventional physicians, so this was a non-randomized comparative study. And there was also a homeopathic arm, which was also compared to conventional medicine, matched for countries, and which was published separately, with similar results. Now, notably, the 27 physicians of the anthroposophic arm, they prescribed 265 different products for these patients with 5 chief complaints. So this illustrates the multitude of drug therapy options in the whole medical systems, like also homeopathy, which makes it so different to conventional medicine. Among the results, the strongest differences pertain to use of antibiotics and analgesics, which were much less used in the anthroposophic arm, here represented by high odds ratios for non-use. improvement and recovery was on the whole somewhat faster among children and broadly similar in adults. The study was not randomized, so recovery may be related to, amongst others, motivation factors which we couldn't adjust for. Otherwise, we did the homework adjusting for baseline differences. But the differences in use of antibiotics and analgesics are so large that they cannot be explained by confounding, and they were also found similarly for homeopathy. Now,